Maybe later.
All right, everyone. Good morning, everyone. I'm Hiro Tanaka from the National Institute of Information and Communication Technology. It's my great pleasure to chair the first part of the morning session. In this part of the session, we have 80 minutes and 2 speakers. I'd like each speaker to finish in 30 minutes and leave 10 minutes for open discussion. The first speaker is Professor Susumu Goto from Kyoto University, and the title of talk is Network Perspective in Bioinformatics. Professor Goto, please start.
Thank you for your introduction. And good morning. First of all, I'd like to thank Murata-san to invite me to this Interdisciplinary Symposium. And my name is Susumu Goto from Bioinformatics Center, Kyoto University. And my research interest is bioinformatics and the network analysis in terms of the molecular or protein-protein interactions or metabolic pathways. So today I'd like to mainly talk about 2 topics which we have done in our laboratories regarding the network analysis. So because I'm from the bioinformatics field, I should start something, a brief mention about bioinformatics. And one of the most important backgrounds that the research field of bioinformatics has come is that we have a large amount of data, especially from the omics experiments, including genomics, transcriptomics, proteomics, interactomics, metabolics, and so on. And these are the the 4 sets of information, including in the cell or organisms. And this information is so large, so it is difficult to interpret the meaning of the data only by the human eye or manually. So we have we need to develop the many techniques for interpreting such large amount of data and solving the biological problem. So in this case, mainly on the molecular level, which means DNA from genomics and the RNAs for transcriptomics experiments and proteins or protein interactions or metabolites for the chemical compounds. And of course, this biological problem includes the elucidation regarding what is life. So, to tell the truth, I do not have many things about to tell what is life, but probably life means something like the autonomy or interaction with others. For example, we are interacting with the others and the environment and the other species or organisms. But we are interested in the molecular level, especially from the data of the molecular biological experiments. So, in terms of the molecular level, so the interaction with others include the, for example, the uptake of nutrients from outside of the cell, which leads to the metabolisms in the, for example, cells, and these cells produce the chemical compound which is necessary for the following process, and also produce the energy for other process. And another thing with interaction to outside is sensing signals from outside, which is also from the environment. And we should, or some biological unicellular organisms should sense a signal for move to the other side or to the forward side. So we need a signal transduction which transduce the signals from the outside and to the a downstream gene transcription or something like that. And regarding the life, probably the application is also important, but I do not mention about this in this talk, but the energies and the signals for those replication is also closely related to this kind of network. So this is one example of the network I would like to mention or use in our research. So this is a metabolic network, and this is originally bearing a wall chart, which is downloaded from the X-Passy site. And we have many chemical compounds and proteins which works in this kind of network, and they are connected in each other. We also have a signaling network. This is also the data from the database called Biocarta. So in this case, these are the proteins and these are the chemical compounds. So we have many protein-protein interactions and protein compound interactions. And those kind of interactions, this is not only the protein or molecules itself. We need to elucidate whole network of these interactions of these molecules. So this is a very schematic view, but so Genome Project originally started to elucidate the list of the gene catalogs and list of the genes to reconstruct the molecular networks I mentioned before. So as a result, Genome Project produced a large amount of data. In this case, list of the whole genes in an organism. And we need something like a synthetic approach from this set of genes to this network. But in reality, it's still difficult to directly go from here to here. So one of the reasons is we have many genes with unknown function, even we have a genes itself. So it means we know the nucleotide sequence or amino acid sequence or even the protein structure, but we do not know what the genes or proteins actually function in a cell. So we need to predict or infer the function of these genes before going to this, or we can use some part of this to infer the function of this unknown genes of unknown function. More specific example is this metabolic pathway. So this is a region degradation pathway of this bacteria Pseudomonas aeruginosa. And we can reconstruct the resin degradation pathway by the genomic information, which is a list of the genes and the predicted gene function. And we noticed that we can reconstruct this part, so colored part. So this part, we know this Pseudomonas aeruginosa actually have this network. So from the genomic information, we can infer this species can convert this compound to this compound and to the TCA cycle. But only from the simple genomic view, we don't know whether the resin can be degraded using this pathway or this pathway by this species, because we don't know the corresponding genes to the enzymes. But in contrast to the genomic information, we have accumulated knowledge on the biochemical pathways, which is not always connected to the genomic information. So in this case, the enzyme listed here are known to exist in this bacteria by the traditional biochemical experiments. But none of their genetic or genomic information are not elucidated yet. So we don't know the correspondence between these enzyme reactions and the genes which catalyze it. So, as the first part of my talk, I'd like to introduce one method to infer this kind of missing part of the network. And because we have several information from the related to the genomic information. For example, we have a gene location. We have a whole genome of this organism already. So we know how these genes are located on the genome actually. And in case of the bacteria, sometimes the functionally related genes are located closely on the genome. So we know if the genes on the chromosome are located closely, there may be some functional relationship among those genes. We also can construct a phylogenetic profile, which is a bit vector of the vector of the zeros and ones for each species, including the current species, which is a Pseudomonas aeruginosa. And if each gene, we can set the 1 or 0, in which the 1 means this corresponding species. So this each column means one species. So this corresponding species have these genes, but the species corresponding to the second column doesn't have these genes. So we can construct this kind of vector, which means that each species have or not have or do not have this gene. And this pattern reflects the evolutionary relationship of the genes. So if the two genes have a similar or same pattern, there is a chance to have some functional relationship. And of course, if there is a genes closely located on the pathway, they have a related function because these enzymes or genes all have a function related to the lesion degradation. So we can somehow correlate this kind of information to the pathway or network information. To do that, we have devised a method to project the original similarity space to the other feature space. And we can define some similarity between these genes using this, for example, phylogenetic profile, and we plot the data. In this case, a circle means a gene, according to the similarity between them regarding the, for example, phylogenetic profile. But the actual network, which means in the gene network or metabolic network, sometimes different. For example, these genes are very close in the phylogenetic profile, but they are not connected to the pathway. There is a case such a thing. So we define some function to map those data into the other feature space, reflecting the actual network process. By using this function, we can also plot other data, in this case genes with unknown function, and to see whether these data are mapped to in this feature space. So, so that we can link this test data to the actual data. So using this kind of methodology, we have predicted enzyme genes corresponding to the missing part of the lesion degradation pathway I showed before. So I'm sorry, this is upside down, but for each reaction, there are 6 reactions from the lesion to the glutaric coil in the missing pathway or network, and we have assigned a candidate gene from the Pseudomonas aeruginosa to each reaction, and actually we have verified these 2 genes by the biochemical experiment, and to see these genes actually function for this enzymatic activity. So in this case, the first function assignment is relatively general, and we don't know the specific substrate, in this case, this kind of compound. So we can, in this case, we could assign the specific compound to this enzymatic activity as a substrate. So to do that, this kind of thing, the network is very important. So the Barabashi from the Notre Dame University proposed the notion of the network biology. So network biology generally means analyze the biological network topologically. So in this case, there are several kind of representation of the biochemical network. This is a chemical compound, this is a reaction. And usually the node degree, which means how many edges exist in each node. So, so in this node degree distribution is followed powerful than the scale-free distribution like this. And he proposed the robustness of the network partly comes from this kind of scale-free network, because the many nodes have only one or two edges, and the very few nodes has a large amount of edges, and this very few nodes are very important. So random attack destroys this part, which does not affect the whole network drastically, but this part is essential. So this kind of network is a source of the robustness. And they also proposed the evolutionary model of this kind of network, which is a preferential attachment model, or rich get richer model. So the first, so some nodes have a tendency to have many links or degree. It has much more edges or interacting partners in the future too. So this kind of topologically analysis of the biological network is so-called network biology, and it is basically to elucidate how biological network has emerged. And the target of the network is sometimes network including the activation or inhibition regulation, so that we can extract the network module as a functional sub-network of the whole network, or signaling the transcription network. And we also have a domain combination network, which I would like to show you as a second topic of work. So the domain combination network means like this. So the protein, in this case the amino acid sequence, has usually several functional or structural domains, so this one protein has several distinct functions, like a binding domain or catalysis domain or something like that. So we can connect the domains if these two domains are on the same protein. So we have we can define the co-occurrence, domain co-occurrence network like this, or domain combination network like this. So in this case, so the domain A and domain B are next to each other, and domain B and domain C are next to each other. So this kind of information described in this network. And we counted the domains or domain combination like this in the completely sequenced or rough, partially completely sequenced eukaryotic species, and their inferred ancestors, like vertebrate or animals or something like that. And we have basically the animals or vertebrates or higher animals have many query specific domains or domain combinations. But as a we can see the network as a topologically, and this domain combination network shows also a scale B property. So we can plot the degree of the domain and the frequency. We have also this kind of power law distribution. And so following the Barabas's model, this network is produced by the preferential attachment model. So all the domains are important in terms of the creating a new function. I mean the combination of the domain. But the once we see the average number of the combination partners for each domain, actually all the domains have more partners, like a preferential attachment model. So in this case, this is a extant, the current species, and this is a very old, ancient species. So if the line like this, the older domains have more partners. But there is some exceptions, especially for the animal point for the animal or invertebrate or vertebrate species has many partners for the domain. So this we believe correspond to the functional explosion of the genes of the eukaryotes, or especially for the animals. And actually, these domains have a very important animal-specific or deuterostome-specific domains. So these are the very brief introduction of our work regarding the network in bioinformatics, and I'd like to emphasize the importance of the network in bioinformatics analysis. And I have talked about the functional prediction use and the domain network analysis using the network. And we also have done something like some extraction of the functional modules in metabolic networks in this article. But it's not so directly related to the question of what is life. And we do not know the answer yet from those analyses. But to know what is life, probably it is important to know what is not life. So to this kind of comparison, we can compare the biological system. Actually used, for example, a domain network or protein interaction network in the biological system, and those not used in the actual network. So to analyze such thing, we need many kinds of information, especially from the genomic or other omics experiments. And we also need a database to store this kind of information to analyze for easy analysis of this kind of data. So currently we have a very standardized database for the nucleotide or amino acid sequence or protein 3D structures. But they are still developing stage of the expression, messenger RNA expression database or SNP typing database or protein interaction. And it is also necessary to construct the knowledge base on the biology or medicine or biochemistry, which is only stored in the literatures. So to construct this kind of database and integrate them for the future analysis, we can actually probably do the network prediction or prediction of gene function, and this leads to the ability to understand life or evolution or simulation. Okay, so this is a collaborators mainly from our laboratories, but these three are four researchers from the experimental field, biological experimental fields, and others for the bioinformatics or theoretical part. And thank you for your attention.
Thank you very much, Professor Goto. That was a very insightful and interesting talk. So now the session is open for comments and questions. Does anyone have any comments or questions? Please.
I'm also a bioinformatician, and I used to study the function of proteome and to predict their functions. And so in the recent protein database, still 50% of the protein is functionally unknown. And the main reason of that is because every species has its own species-specific protein, and which also its functions are still unknown. So can you reveal these species-specific orphan genes and the function of orphan genes by using these kinds of analysis?
For the first part, it's still, of course, difficult, but the well, in this case, if in this case we somehow know the type of the enzymes, which means that there are low similarity to the other genes from other species. So in this case, it's probably a bit easier, but we have other example from here, and I didn't mention. So in this case, we do not use the network analysis, but in fact, this part is predicted by different approach, which means in this case, we have a knowledge on the amino acid composition of the enzymes, but not the whole sequences. So in this case, we can search the amino acid sequence, genomic information using the amino acid composition. So we, if we have such information, we can assign somehow the genes to the current enzyme, and this is not. In this case, we do not need any similar proteins from other species. But in general, it's of course still difficult.
Is there any comments? Question? Comment?
Thank you for your presentation. And I was interested in mention to the domain partner analysis. You mentioned that something's occurred with a combination of function explosion you mentioned. I found two peaks in that. On the second peak, maybe related to the appearance of vertebrate.
Yes, actually, it's animal.
But how about the first one?
This one is a bit difficult because this is actually eukaryote. So this is a distinction between the eukaryote and the prokaryote, which means that sometimes can relate it to the multicellular and unicellular something.
So it almost correspond to the Cambrian explosion, much more earlier stage.
Probably this is much more earlier stage. So I don't know the Cambrian stage, but the it's something like here. I think. Actually,
I think that that functional explosion may be somehow related to the gene duplication.
yes. Right.
So probably more earlier than that. So my question is, why these exploring domain domain increase, but
Yes.
the main partners increase?
Yeah. I don't know the mechanism yet, but the to examine the detail of the function of the domains here, we see many protein kinase domains or other domains which is specific to the signal transduction in the animals or other mammalians or something like that. So this means, I'm sorry, this discussion is here. So this vertebrate has a, I forgot, but the either to have a function of the protein kinase or something like that, and the other one has a, probably this one has a immune system specific domain. So I think mechanism is still unknown, but some important functions for these specific species have explored in this time. And as for the Cambrian state, we cannot, we don't have any species which have a Cambrian state, Cambrian eras species in the current completely sequenced species. So we have, if we have many more completely sequenced species, maybe we can subdivide this part and have more detailed results.
Thank you for a very nice talk. I'm working on the almost same problem, but not informatics point of view. I am a theoretical physicist to study mathematical part of this problem. As far as I know, in scale-free networks, the exponent gamma you showed is the values for various types of species.
Yeah.
I think gamma. If gamma is 2.5, that is a critical, So, do you have some indication for the difference between below gamma equal 2.5 or gamma is greater than 2.5? That's significant because network structure is very different even in the system scales free. Gamma, if gamma is less than 2.5, the system looks like there are several half size which dominates other ring size. But if gamma is greater than 2.5, like gamma equal 3, that is a Barabash model. exactly. That's interconnected, even in scale 3. So there is a very difference of geometry.
Yeah. In fact, this is a very strange species in terms of the genomic information, and sometimes the functional annotation itself is the gene annotation, which means the assignment of the genes in the genome, is still not so correct. So I think this is probably the exception, and we did not check the reason why the decay, but maybe we should check. Thank you.
Okay, I think we have used all the time for the first speaker. Let's thank the speaker again.